Hello everyone, my name is Rahul. In the previous video, we had discussed about Golgi apparatus. In this video, we are going to discuss about lysosomes. So lysosomes are synthesized by Golgi apparatus along with endoplasmic reticulum. However, the main synthesizing organelle is Golgi apparatus. However, whatever the material is present inside the lysosome, it is synthesized by endoplasmic reticulum. So all the enzymes that are seen inside the lysosome, they are synthesized by endoplasmic reticulum. Lysosomes are also known as suicide bags of the cell. Lysosomes are also called as suicide bags of the cell. Why are they called so? We will see in a short while. Now, lysosomes, they are membrane bound. These are membrane bound vesicular structures formed by the process of packaging in Golgi apparatus. The isolated lysosomal vesicles have been found to be rich in almost all types of hydrolytic enzymes. So these are rich in almost all types of hydrolytic enzymes which are optimally active at a pH of 5. So they are optimally active at an acidic pH. These enzymes are capable of digesting proteins, carbohydrates, lipids, nucleic acids, etc. So these hydrolytic enzymes, they can lyse or they can hydrolyze any of the given macromolecules. So except mammalian RBCs which do not have any organelle, all cells contain lysosomes. So except mammalian RBC, all the cells contain lysosomes in animals. These lysosomes are seen less frequently in plants. They are seen less frequently in plants. Now, lysosomes are covered by a membrane which is similar in structure to the cell membrane. So, there are almost 50 types of hydrolytic enzymes in each lysosome. So membrane of lysosome has an active H+ pumping mechanism which produces acidic pH in the lumen of lysosome. So if this is supposed to be a lysosome, then the membrane of the lysosome, it continuously pushes H+ inside by the use of ATP. So by an active process, it is continuously pumping in hydrogen ions, thus creating a acidic pH which is optimal for the functioning of lysosomes. Lysosomes are highly polymorphic. So they can be seen in various shapes and sizes because they have different physiological states, they have different functions. At different times, they are performing different functions. Now, moving on to types of lysosomes, we have the primary lysosomes, which are also known as storage granules. They store enzymes, which is mainly acid hydrolase, that is hydrolase, which is active at an acidic pH in inactive form. In inactive form. Only when the cell will need the enzyme to be active, the enzyme will be activated. Otherwise, this is present in an inactivated form. The next are known as digestive vacuoles or heterophagosomes. They are formed by fusion of primary lysosomes and phagosomes. Digestive vacuoles or heterophagosomes, they are found by fusion of primary lysosomes and phagosome. So whenever a cell it is going to ingest a food particle, there is going to be formation of a phagosome. This phagosome it is going to fuse with the lysosome and thus leading to the formation of phagolysosome containing the food particle inside and the presence of digestive enzymes also inside this heterophagosome. Next are known as residual bodies. They contain undigested material. They contain undigested material and eliminated by exocytosis, that is movement of a substance outside the cell. Next type of ribosomes are known as autophagosomes, which are also known as suicide bags. These are lysosomes that are used to digest the worn out or damaged cell organelles within the cell. These are known as autophagosomes. Now moving on to the functions of lysosomes. They perform the function of intracellular digestion, which may be heterophagy or autophagy. In heterophagy, it is going to digest foreign materials received by the cell. Example, food particles, bacteria, etc. So any microbe that is entering the cell, it can be digested by the lysosome. Autophagy means digestion of old or worn out organelles, and since it is digesting the cell's own organelles, this is known as autophagy. They also perform the function of extracellular digestion, such as lysosome of osteoclast, which is the bone destroying cell, dissolves unwanted parts of bone during bone remodeling. So the bone is continuously being remodeled, it is continuously being made and destroyed. So lysosomes play an important role. Now cellular digestion, which is also known as autolysis. During autolysis, lysosomes burst. This leads to release of hydrolytic enzymes, thus causing digestion of entire cell along with plasma membrane. So the entire cell is going to be get digested. This is why autolysis is also known as suicide by the cell, and thus the lysosomes are known as suicide bags of the cell. With this, we finish off with this discussion on lysosomes. See you in next video.